స్టార్ట్ చేసేద్దామా సార్? Dear esteemed participants, respected faculty members, a warm and vibrant morning to everyone as we gather virtually for the third day of our enriching one-week national online faculty development program on sustainable innovations in drug delivery, a multidisciplinary perspective. Organized by Department of Pharmaceutics and Biotechnology at Kvsrscops College of Pharmaceutical Sciences in association with the Indian Pharmaceutical Association Siddhartha Nagar branch. This FDP has brought together academicians, researchers, and industry professionals from across India to share insights, strategies, and breakthroughs in innovative drug delivery. I extend heartfelt thanks to our organizing committee, invited speakers, and all attendees for making this event a progressive success. Wishing you an inspiring and intellectually rewarding day. And now, it's my privilege to invite Dr. G. Ramana Sir, Professor and Head of the Department of Pharmaceutics and Biotechnology, and our esteemed Program Coordinator to the virtual podium and commence today's session to guide us through a stimulating day of learning and exchange. And introduce our today's session resource person, Dr. Surya Prakash Rao Kovasu Sir. Over to Dr. G. Ramana Sir.
Yeah, thank you, Arifa.
Thank you, sir.
Yeah, good morning, everyone. It gives me immense pleasure to introduce our distinguished resource person for today's session of the Faculty Development Program. He is Dr. Surya Prakash Rao Kovasu. Happily to share that he is our alumni student. He passed the B.Pharm in the year 2005. So we are honored to welcome Dr. Surya Prakash Rao Kovasu, a respected expert in the field of pharmaceutical research. He currently serves as Senior Principal Scientist at Pedagis LLC, New Hope, Minnesota, USA. Dr. Surya Prakash has made significant contributions to the field through significant publications, patents, projects, regulatory achievements, and collaborations. He has been actively involved in training, mentoring, and capacity building within the academic and industrial settings. Today, he will be speaking on formulation of dronedarone hydrochloride loaded proliposomes, in vitro and in vivo evolution using CACO2 and rat model. He has vast experience in all phases of parenterals, include peptides and small molecules, solid oral, liquid oral product development for 505B1, 505B2, 505J regulatory pathways. He is also expertise in formulation development of lyophilized products, poorly soluble drugs using proliposomal, liposomal, solid lipid nanoparticles, and self-emulsified drug delivery technologies. He has expertise in pre-formulation, formulation development of long-acting injectables, drug-device combination injectables, ophthalmic suspensions, control-release liquid and solid orals. Dr. Surya Prakash previously worked with Agility Pharmaceuticals, Ambioform Incorporation, and Agent Drug Research Solutions Private Limited. Coming to the academic credentials of Dr. Surya Prakash, he completed his B.Pharm from Kommareddy Venkata Sadasivarao College, Siddhartha College of Pharmaceutical Sciences, M.Pharm and PhD from Andhra University, and postdoc from Western University of Health Sciences, California, USA. Dr. Surya Prakash was awarded with UGC JRF in Engineering and Technology during 2009 for doing a PhD during 2009 to 2011. Qualified GATE in the year 2006 and awarded with the AICT sponsored fellowship. He has also won first prize at AICT sponsored national seminar on particular drug delivery systems during 2011. He also received Swarnapatra Puraskar, a state level art competition on the occasion of 50 years of Independence Day in August 1997. Again, his recognitions approved from US Citizenship and Immigration Services, and the government, US government is approved O1A during the year 2019, and also he approved EB18 in 2023, and other professional recognition from the scientific field. He published more than 30 research publications in various international peer-reviewed articles, journals. And also his citations is more than 500, H index 11, I-10 index 15. He is a speaker at national and international conferences and judging the work of the other professionals in pharma sector in the 124 occasions. A technical advisory board member, SDS Incorporation, editorial board member for many journals. He presented many poster and oral presentations at the national and international level. So now I request, please join me in giving a warm welcome to Dr. Surya Prakash for his talk. Welcome, Surya Prakash.
Thank you, sir. Thanks for your nice introduction. I'm happy to see my guru here, and also I'm happy to see my gurus as well, like other gurus like Kanasri Madam, Bharti Madam, and other colleagues as well. It's my pleasure to speak in front of all my gurus, and also I'm happy to deliver this topic today. Thank you once again, sir.
Thank you.
Yeah.
Surya Prakash.
Please, can you? Hi, madam. How are you?
Fine. How are you?
I'm doing good, madam. Happy to
Nice
see you
to
all.
see you.
here.
Very nice to see you.
Thank you, madam.
Okay,
Please,
okay.
can you confirm my screen, madam? Oh, sorry.
Yes, sir. Your screen is visible.
Just a minute. Okay, so hope everyone can see my full screen. Dr. Ravi Garu, please can you confirm my screen full screen?
Yeah, nice,
Yes,
Surya
sir.
Prakash. You can continue.
Yeah, thank you, sir. Good morning, everyone. It's like a good night for me, but I will try to give my best for this presentation. Coming to this presentation, I would like to talk about formulation of dronedarone hydrochloride loaded proliposomes for in vitro and in vivo evaluation using CACO2 and rat model. So I would like to present this as a case study one. This is a research publication. Followed by this one, I would like to present case study two, like how it impacts like kind of an IND and NDA submissions. So moving to this presentation, just a minute. It's actually blocking my screen. Yeah. Product approved in Orange Book. So this topic is like, I would like to, before going to the topic, I would like to discuss about a couple of liposomal products and as well as proliposomal products. Liposomes like kind of recent technology. We can say like the first product is approved in the year 1995. Ebelset amphotericin B is like first product where they used DMPC as the lipid. And when we see these liposomal products available in the market, there are like a couple of liposomal products available in the US market and also Europe market. And they are varied from anti-cancers to pain treatment. And these liposomal formulations like doxil, deposite, onivide, Vixos, these these formulations are launched in Orange Book in the recent years upon the years like from 2012, 2015, and 2017. And also we can see a couple of products, other products like Exparel, Ambisome, and Myoset, Vioxys. The criticality of these liposomal formulations is like selecting a proper lipid carrier, as well as understanding its liposomal formation, whether it's a hydrophilic drug or a hydrophobic drug, whether it's lipophilic or a hydrophilic. And also we have to carefully go through this BCS classification and log P values as well. Because if it is a water insoluble drug, its formation will be different. If it is a water soluble drug, its formation will be in a different composition. So when we see these log P values, especially minus values, negative values indicates values near to zero indicates high water solubility. And log P values higher, like higher numbers or positive numbers always indicates like kind of lipophilic drugs. So we have to better understand what kind of lipids we have to use here, as well as the formation of the liposomes. In these liposomes, we can see like small unilamellar liposomes, which are like size of 100 nanometers. Large unilamellar liposomes, which are a size of like 200 nanometers to 800 nanometers. We also have MLVs, we call it as like multi-lamellar vesicles, which are from like 500 nanometers to 5000 nanometers. Generally, we don't prepare these MLVs until unless there is a high drug load and larger molecules. And also a couple of other liposomes like long circulating liposomes and targeted liposomes. And these targeted liposomes, we can even deliver antibodies and they will specify and bind to the, adhere to the antigens as well. So these liposome technology is increasing drastically day by day and significantly. When we see the like the image far bottom, the left side, in 2018, the market value is 3,600 US million dollars. Currently, like if you consider it as like 2002 to 2007, it is between like 4,000 to 6,600 million dollars. It is projected to almost like $11,293 million by the end of 2032. This indicates that the significance and the increase of liposomal formulations in the market and also target specific, less toxic and less side effective formulations, the need of liposomal formulations. So considering all these parameters, majority of the pharma companies, they are focusing and they are trying a lot of research in phase 1, phase 2, phase 3 studies, that is for NDA developments. But on the other side, there are a lot of complications, especially its stability, as well as its aggregation properties and its storage conditions. Along with these conditions, we can even have a lot other complications like encapsulation efficiency, commercializations, and also achieving the therapeutic efficacies. Coming to these storage conditions, most of these liposomes are stored at like 2 to 5C condition because of its poor storage conditions. And its accelerated conditions are generally indicated as like 25C conditions, but couple of liposomes can be stored at like higher temperatures. If you see all these liposomes, I have given the images of these liposomes at the bottom. You can see these vials as a lyophilized products. Preparing liposomes is one difficulty, and further formulating these liposomes as lyophilized products is another difficulty, because like commercialization, scale up of lyophilized cycles are much more tougher, and also the consistency also requires a lot. Apart from this one, agencies requirements like extractable, leachables, all these factors come into picture. So to address all these issues, most of the researchers and most of the companies currently focusing on proliposomal formulation. This proliposome term first time coined by Payne et al. In the year 1986. These proliposomal are slight modifications to liposomes, where liposomes are available as like liquid phase. Further, we have to add the like carrier systems like mannitol and MCC to further do these. Mannitol to further prepare these lyophilized products. But in case of liposomes, there is no need of preparation of any lyophilized product. Simply to these liposomal formulations, we can add the carrier systems like MCC, mannitol, etc., to make it as a solid dispersion, kind of solid free flowing powder. This free flowing powder can be tabulated and it can be filled into capsules, and we can further control the release by different types of coatings as well. So that means by preparing these solid proliposome formulations, we can see a betterment in the stability as well as dose administration, because all these liposomes, which are available as lyophilase products, are intended for injectable route, which need to be taken care under the supervision of doctors. But in case of proliposomes, the technology is such a easier and such a handy that it can be formulated as tablet and capsules, and which can be taken very easily. So considering all these parameters, we have focused our research on proliposomes. Our lab is majorly focused on proliposomal technology at Western Center for Drug Development under the guidance of Dr. Guru V. Betagiri. He has multiple patents, and as well as we published a lot of papers on different formulations, categorizing from BCS class 1 to class 2, class 3, class 4 as well. So in this current paper, we considered developing proliposomes of dronedarone hydrochloride, which is a poorly soluble drug, comes under BCS class for low solubility and low permeability. And this drug is used for antiarrhythmic drug, and it works by blocking multiple ion channels in the heart, including sodium, potassium, and calcium channels. If you see the chemical structure, it consists of methane sulfonamide hydrochloride, and its molecular weight is 593. It's white to off-white in color. Its melting point is approximately 144 C. Its log P value is 7.05, which is highly lipophilic in nature. And its solubility is soluble in chloroform, but is poorly soluble in water. We have selected this dronedarone hydrochloride as a drug candidate for our research. And for ease of convenience, I'm going to call it as DRD. So DRD means dronedarone, the code which we have given for ease of pronunciation. And coming to the reference product, before we start the research in the lab, we started digging the information from the reference product from the Orange Book. When we see this information, this project is actually started in the year 2016 in the mid of May and June, around mid of May and June. And when we started doing research, we have noticed that there is a FDA filing in 2009 for ANDA by Sanofi Aventis. This is by type 1 new molecular entity. And this NDA filing come into picture from July onwards. The indication is cardiac arrhythmias. The mechanism is not clearly known, and it belongs to amiodarone group. And coming to its bioavailability, it is only 4% bioavailable. When it's taken along with the meal, its bioavailability can be increased up to 14%. And when we see the strength, the tablet strength is 400 mg. Most of our tablets, like 50 mg, 20 mg, 30 mg, like that. If you see like one of the example, like fenofibrate, when it is marketed, like in early 2000s, its strength is around 200 mg. But for further solubility enhancement and further permeability enhancements, the drug's strength was drastically decreased to 200 mg to 160 mg, further to 120 mg, and further to 60 mg. I think currently 30 mg tablets are available. So considering such kind of examples, if you see this dronedarone, which is 400 mg of strength, it's a too high strength because of its low oral bioavailability. And also we noticed that it's 98% of its drug bonds to the plasma proteins. Because of this plasma protein bond, very less quantity of drug is available to reach the site of action. And plasma protein binding can restrict the distribution of drug to certain tissues and organisms. That's why we have seen very low concentrations of plasma. And because of this plasma bound proteins, it's a general theory that its elimination will be higher. So that's why we are seeing almost 13 to 19 hours of elimination. And also by this approach, by selecting these liposomal formulations and further formulating it as proliposomes, we are planned to increase the solubility, further to enhance the permeability, and further we want to study this product in vivo. So because of these liposomal formulations, we anticipate that definitely it will circumvent this pre-systemic metabolism. We all know that the first pass metabolism, that is like lipid formulations through transcellular and paracellular route enter into cells and forms lipoproteins and chylomicrons, which are transported by the intestinal lymphatic system and thus by bypassing the first pass effect, resulting in improved oral bioavailability. So this is the mechanism actually involved in these liposomes. So we, based on our prior experience, based on the prior research, we were like aimed for seeing these avoiding this pre-systemic metabolism, and by doing this proliposomal technology. So the approach here we are following is proliposomes, which are useful to enhance the transport of drugs via lymphatic route. And these proliposomes can be formulated as tablets or capsules, just now we discussed. And also further, once it like in contact with water, it forms the multilamellar vesicles or unilamellar vesicles. So given this brief introduction about the liposomes and proliposomes and need of liposomes and its market value, and also the drug candidate and the reference product, then we started doing our research. So we started with these materials and methods. This particular presentation involves materials, preparation of proliposomes, analytical methods, proliposomes evaluation, like liposomes particle size, zeta potential, encapsulation efficiency, assay of dronidone capsules, in vitro dissolution studies, in vitro permeability studies by CAC2 model, cell uptake studies using rhodamine, DSC studies, as well as plasma estimation using LC-MS method, pharmacokinetic study, and stability study. So in this paper, we majorly focused on proving the in vitro dissolution studies as well as in vitro permeability studies to select a suitable and right formulation, which is giving a 100% success rate during the in vivo studies. So for this one, we incorporated multimedia dissolutions as well as in vitro permeability studies by CACO2 cell models. This is the novelty of this research paper. Also, the novelty of this research paper is by the time there are no dronidone liposome formulations available or published in the market. So given this one, just I'm going to the materials. It's like big lengthy summary here. You can see a lot of lipids, DMPG, sodium, HEPC, SPC, soy phosphatidylcholine, phospholipam, and some other lipids are also evaluated as part of preliminary screening, but we didn't present those data in this publication. And the best performed lipids are captured in this presentation. And cholesterol is also part of this study, MCC as well as perlitol. And dronidone is procured from BOC Sciences. The major study, actually, we have seen a lot of complications during this research is doing the CACO2 studies. Maintaining the cells and procuring the materials, preparing the medias, and doing the studies. It took almost 3 months for us to perform these CACO2 studies in parallel to the all other developments. In this CACO2 study, you will come across around this DMEM media and PBS, DPBS, and fetal bovine serum, FBS, EDTA, HBSS, and 6 well plates. Also, we have procured pre-cannulated Sprague Dravely rats. We used to call it as SD rats from Envigo, San Diego, and also all other solvents and excipients from EMD Millipore. I would like to give you a brief introduction, like a brief, summary, like how these liposomes are prepared by. If you see this image, actually, I have prepared this image like a couple of months ago by using BioRender. And also even the first table I have prepared for this presentation. I'm happy to share all this information with the team. And when we see this liposomal formulation, liposomes are proliposomes. Generally, like drug and lipid will be taken into organic solvent, and we have to ensure for a clear solution. In case of proliposomes, once it forms a clear solution, adsorbent, MCC or mannitol, whatever it may be, we can add that one, and then keep this RBF, round bottom flask, to rotavapor. We used Wookie rotavapor, and then dry the thin film. Once the thin film forms, in case of liposomes, it should be hydrated with water, which forms multilamellar vesicles as well as unilamellar vesicles. Most of the times you see multilamellar vesicles. Usually what we used to do is pass these multilamellar vesicles to the extruder, some of the dialysis membranes to get the unilamellar vesicles for having the content uniformity. In case of proliposomes, the dried thin film, which is consists of MCC, drug plus lipid, it can be scratched and collected and then passed it to sieve number 200, sieve number 60 mesh, that is 200 micron size, and get the uniform powder. That powder can be filled into capsules or further can be compressed as tablets. And also, just I would like to present a conventional liposome image here. Most of the liposomes, conventional liposomes, they have two layers. One is hydrophilic inner core layer and outer lipidic bilayer. So aqueous drugs will be embedded in the inner core layers, and the lipidic drugs will be embedded in this bilayer, lipid bilayer. So our drug, dronedarone, is embedded in the lipid bilayer. So this is the general information about the manufacturing process. And also here we have to consider one peculiar parameter, that is phase transition temperature, as well as we have to consider the glass transition temperature. These are the temperatures where the lipid gel converts to crystalline state. That means rigid to fluid state. Temperature at which it occurs called as melting temperature. That's why it given as TM or MP. When we see this DMPG sodium, its its melting temperature is around 23 degrees. It can be increased up to 41 C. And in case of phospholipon, it is 51 C. But soy phosphatidylcholine and other one, HEPC, it is in minus negative values. And also there are a couple of other products like DSPC is 55 C, DPPE is 63 C. So there are different examples. We have to consider these parameters while selecting the liposomes preparation. Coming to the key table, that is formulation, composition, and physical characterization. When we see this table, it clearly gives what we have done here. It's summarized here. We have selected FPC, soy phosphatidylcholine, phospholipon, 90H, DMPG sodium, HEPC. When we see this table, the basic information we are getting here is the drug to lipid is to cholesterol ratios are given in parts. First, we tried with SPC, and in case of SPC 1:1, 1:2, 1:4, we have achieved highest entrapment efficiency with 1:4, that is 65. But it's not so significant. Coming to the particle size, it is ranging from 200 to 340 nanometers. Then we started using phospholipon 90H with 1:1, 1:2 ratios. The entrapment efficiency observed to be 54 to 67. Then we moved to DMPG sodium, 1:1, 1:2, 1:2. Here we used to get very good numbers, that is 62 with 1:1 ratio. And also the particle size is 348. And coming to the 1:2 ratio, 1:2 ratio, it is 132, and then entrapment efficiency went up to 79%. In case of FET formulation, what we did is we added cholesterol to increase the rigidity. So by increasing the cholesterol, the particle size was slightly increased, but the entrapment efficiency was also increased to 84%. On the other hand, we tried HEPC, which is also not giving a good data. But overall, by seeing this data, physical characterizations, we have selected DMPG sodium as the next formulation for the next studies. So here I would like to give you a small note to the team, note to the audience, that encapsulation efficiency is very tricky, because most of the times we use regular FNWs to collect the samples, and we overestimate the entrapment efficiency. We used to see a lot of research articles saying 90% entrapment, 80% entrapment, but that's not true, because we have to select a suitable molecular weight cutoff, that is 30K, KDA, or 20 KDA, 15 KDA, 10 KDA, 5 KDA. So this molecular cutoff indicates that particular Daltons, number of that particular Daltons will retain on this molecular sieve, and the remaining will pass through it. The remaining means obviously the free drug will pass through it. So we can see how these these cutoffs will look like. So here in the small pouch, we can keep the drug solution aliquots. And once we centrifuge it at a different RPM, 2500 RPM or somewhat, and it will separate free drug and also the drug with liposomes, because liposomes are in bigger particles. Once we have this free drug, it can be further diluted with acetonitrile water or any methanol combinations, and further it can be estimated. So finally, we can estimate like total drug, like the equation is total drug minus free drug times 100 divided by total drug. This gives encapsulation efficiency. So choosing a molecular weight, correct molecular weight cutoff is more critical here. And also coming to the PDI was greater than 0.15, and all these formulations indicates that it's a polydisperse system. Considering the time, like I think it's already like half an hour past. I would like to complete the remaining sections mostly in the next 20 minutes. Maybe I will be going a little bit faster. Sorry for this one. And next, we move to the pH-dependent dissolution profiles of dronidone hydrochloride APA as well as Multec tablets. Multec is the RLD reference product. Before doing these dissolution studies, we went to the Orange Book, like we have visited like Orange Book, and we have seen the OGD recommendations, Office of Generic Drug Recommendations. There it is recommended that go for pH 4.5 phosphate buffer using 1000 ml volume and collect the samples till 120 minutes. And also use for paddle with sinker. This is a release media and QC medium. And we did the same thing here. Like what we did here, like you can see the release profiles. Just a minute. So we can see the release profiles. Like we did the multimedia and pH 1.2, pH 4.5 phosphate buffer, and pH 6.8. In case of pure drug, that is API, the release is higher in pH 4.5. That's what actually the release media is. Orange Book is also stating the same. In case of like pH 1.2 and 6.8, we didn't see like very minimal release up to like 10%. In case of Multec, the same observations. Highest release is seen in like pH 4.5, we can see here. And in case of like pH 1.1, 6.8, there is minimal release. So given this information, we have selected the optimized formulation, that is formulation F7 and F8 for further studies. So we can see those studies here. Like here, what we did is from the previous studies, we didn't consider the regular release media, that is QC media. We considered the media which is having lowest solubility, that is pH 6.8. So conditions here are pH 6.8, 1000 ml, 75 RPM, and dissolution up to 4 hours. And also aliquots were like we did for n is equal to 3 samples, and aliquots were filtered using nylon filter of 0.45 before the estimation. And we used a Island 708 DS system. It is a manual, like automated one. The sample collectors is Island 850 DS model, Santa Clara. And all these formulations, SPC, phospholipon, DMPG sodium, HEPC, all these formulations were conducted for dissolutions in pH 6.8. When we see the dissolutions, especially the SPC, we have seen a maximum release of like 20% when we increase the concentrations, lipid concentrations to 1:2. In case of phospholipon 90H, the dissolution was observed to be maximum 10% in case of 1:2 ratio. In case of HEPC, dissolutions are slightly better, but not so significant. It is like almost on 30%, with 1:2 ratio. So in all these cases, we have seen that increase in concentration of the lipid, 1:1 to 1:2, there is a slight increase in the entrapment efficiency as well as slight increase in the dissolution profiles. Coming to the DMPG sodium, which is our interest of composition. So here there is a significant increase of dissolution, especially in pH 6.8. We can see 4 dissolution profiles here. The bottom one, the last one, touching to the baseline x-axis, is the plain dronidone. You see, like it's like kind of almost like 0% release, like maximum of like 5% release. But when we increase the concentration from lipid concentration from 1:1 and 1:2, we achieved almost 50%. Actually, the number of like the dissolution profile 50%. looks lower, but still considering its low solubility of this media, we still like got the better dissolution even with modifying the formulations as like plain drug as proliposome formulation. So given this information, we have selected like the media as pH 6.8, and also we have selected the formulation that is F7. And also here you can see one formulation that is with perilitol. Actually, perilitol is also used as a carrier and as like mannitol. But when we use perilitol, we have noticed that there is a retard in the dissolution. So that's why in the further studies, we avoid using perilitol. We simply went with MCC grade 102. In the next study, the two formulations, that is F7 and F8. In case of F7, the composition is 1:2 ratio. In case of F8, we included cholesterol as well. So cholesterol inclusion is helpful to form this rigidity. And most of the literature suggests to add the cholesterol from the range of like 0.1 to 0.5%. Given the information, we directly went with 0.2% part, and we have seen a significant and drastic increase in dissolution with these cholesterol formulations. We can see like almost a 90% formulations. When we recollect back, the first formulation at 6.8, we didn't see any dissolution profile for the API. But when we further modified these formulations, also further modified this buffer system, especially with SLS, and the volume reduced from 1000 ml to 900 ml, the dissolution was carried for 4 hours, and we have achieved like 90% of dissolution with F8 formulation. So here we came to a conclusion that addition of cholesterol in the formulation resulted in increased drug release, which might be due to altered affinity between the drug and lipid. So with this confirmation, we move to next studies, and that is CACO2 permeability studies. So just to once again give you a brief note that we don't want to rush to in vivo studies just by doing some physical characterizations. That's why we are doing a lot of research, especially on the multimedia, as well as discriminated dissolution and further modifying the dissolutions. After getting all this information, then we move to in vitro permeability studies. So as part of in vitro permeability studies, we have selected CACO2 permeability model. And also there are other approaches like ex vivo studies and also PAMPA models, parallel artificial membrane assays. But we didn't use all those other models for this particular research. When you see this CACO2 permeability models, I started working on these models like late 2016 during the holiday season, like November. So it's like a kind of doing a lot of exercise and a lot of work for these CACO2 cells. So we have prepared the complete media, which consists of DMEM, a BS, non-amino acid, streptomycin and penicillin in a combination. And then we have taken the frozen, the samples like CACO2 cells of passage 40 or something 42, which were frozen in dry ice. We have collected those samples, and when we thawed it, and we started reactivating the cells at 37 C using carbon dioxide of 5% at 90% RH chambers. And the generally it is ideal to start the passage at 40 passage, and doing the actual uptake studies between 45 to 50 is gives a good results. We have considered Uppsala's University of Uppsala, Switzerland's protocol. The lab guys prepared a very nice protocol. We have taken that as a reference, and we followed that protocol with slight modifications considering our lab conditions. And once after thawing these cells and growing in complete media, once we saw this 80% of confluence, and then we started subculturing the cells to the new plates, kind of new these neck bottles. And also these subcultures were done for every 5 days at 1:4 ratio. That means like with a 80% confluence using trypsin and EDTA. What this trypsin and EDTA does here is like they detach the cell layer which is attached to the base of this plate, and we can collect the samples and we can transfer to the new flasks. So by aspiration method, we used to transfer this one. Like this, we did like 4 passages, and from number 40 to 45, once we reach their cells are fully activated. For these 4 passages, it's 4 to 5 passages. It's always almost all taken like a month. Then on the other side, what we did is like we started working on the polystyrene inserts on the day of the actual experiment. These polystyrene inserts consist of like 24 dia and 3 micron pore size. To this one, we added a ragtail collagen to act as a adherent and forms a thin layer on the polystyrene inserts under the micro hood and under UV lights. Once these like polystyrene inserts were coated with rat collagen, these were like dried, and then the suspension of the cells were loaded like using this HBS as a media were loaded to the inserts. These inserts are fitting into the donor receptor plate so that it will becomes as a cup and cone size of kind of setup. And the cell density was used as like 2.5 into 10 to the power 5 numbers, 2.5 times 10 to the power 5, because more the cells actually it won't forms mono layer. Sometimes there will be a high crowding, and lesser the sizes you will see a lot of junctions, lot of gaps between the cell layers. And once we form these incubate these cell layers for like couple of days, then we started exchanging the media on the apical side, on the top side for 1.5 ml and basal side and 2.5 ml. And we started doing this study for every other day. That means for every one day, like today and tomorrow, today day after tomorrow, like that. For every other day, we used to change the media. And after 21 days, it was washed with HBSS and further once again transferred with HBSS. And finally, we started like these plates were kept in the like temperature maintained shaker bath at which is maintained at 37C condition, and for which we started measuring the TEER, like the resistance. Resistance indicates that resistance more than 300 microns, like ohms, 320 or something, gives us that the cells formed are uniform and having good resistance. Lesser the resistance means there are a lot of gaps. So that means whatever the number, whatever the permeation we are seeing, it's not the true number. So after confirming with these TER numbers, then we started taking the keeping the samples, that is sample at a concentration of like 100 like 168.6 micromoles, as well as formulation also in the same concentration, by dissolving it in DMSO to for solubility and further dissolving it in like mixing with HBSS. And these media with drug concentration, API and formulation concentrations were loaded to the inserts, and at each and every time interval, the aliquots were collected. The collected aliquots, like 500 microliters, were tested for further concentrations, and further apparent permeability PAP was calculated using the formulation equation given here: volume into drug of receptor chamber by drug in initial concentration, and that is at donor chamber. So we have seen that P apparent, that means apparent permeability increased from like 1.5 times 10 to the power minus 7 to 1.36 times 10 to the power minus 6. That means there is an increase of like 2.64 fold when we compare the pure drug versus the formulation. And also we have further confirmed that pure drug, the literature data, especially FDA, generates this data to give the BCS classification. This is also one of the study to assign the BCS classification. So we have confirmed these results, especially the pure drug results, with literature available data that is published by FDA. When we use this HPSS at different pHs, we didn't see any difference with respect to the P apparent number. That is also good sign. But when we see this formulation, especially treated with HPSS, we didn't use any DMSO because this is suspension. We have seen like 2.64 fold increase. And from this layer, we concluded that when cultured as a mono layer, CACO2 cells differentiate to form a tight junctions between the cells and serves as a model of paracellular movement, which is almost all similar to our gastric epithelia. So from this one, efflux of proteins, the mechanisms like efflux of proteins and phase 2 conjugation engines models are the pathways which increases the transportation from the apical side to the basal side. And also these cells mimics, as I mentioned, it mimics the human intestinal epithelium. From these studies, we got further confirmation that the formulation selected F8 is further showing in vitro permeability, and we further proceeded for cell uptake studies as well. Actually, we were not planned to do this study, but as we already generated this CACO2 monolayers, we started doing this study as well. For this one, this very quick and simple study, formulation along with the dye, which is like liposomes are formulated along with the API and dye, as well as plain formulation, that is not plain formulation, sorry, it's plain drug plus dye as a suspension. Both were given to this CACO2 medium, which is incubated at 37 degrees, which is having almost 80% confluence, which are carefully washed with the HPSS. To this one, these formulations were given, like formulation and API suspensions were given. And further, it was rinsed with PBS for almost 3 times just to remove the excess dyes and liposomes and free drugs. After that, once again, HPSS was replaced and the images were observed under the microscope. We can see a clear distinct difference between these two studies. If you see the far left one, that is like ACO2 cells treated with dronidone API plus dye. And in case of the right hand side one, which is like proliposomal formulation, you can see a clear cut indicate that is uptake of this dye indication. So these studies also further confirmed that the formulation selected is having higher uptake and cellular because of the cellular internalization of drug. So from all these studies, we further proceeded to DSC studies, where this like cool heat cycle is followed, and the unit used is a TA instrument, and also the RAM temperature is like 10 degrees Celsius per minute. And we can see the pure drug, which is like 139 degrees Celsius. We initially like 143, and also second time I think it is 138 degrees Celsius. Initially, we have discussed that literature melting point is 143. Coming to the formulations, dronidone F8 formulation initial and during the stability, the temperature was, the peak was shifted to 120 degrees Celsius, and there is no change between T0 formulation versus T6 month formulation. The change is actually resulting from physical arrangement of drug from crystalline to amorphous state. And also, as I mentioned, there is no change in the physical, like there is no change in the peaks during the stability as well. So further, we evaluated these formulations for physical parameters and dissolution profiles during the stability, that is at 40-75 condition as well as RT condition. And the powder, almost all quantity of like 620 mg, that is equivalent to 100 mg of dronidone, were filled into size 00 capsules, hard gelatin capsules, and there were 30 capsules are filled into the bottle along with the cotton to absorb the moisture. And these bottles were loaded for stability chambers at 40 C condition as well as room temperature conditions. When we see these dissolutions, there is no change in the dissolution profile at 40-75 condition as well as room temperature condition. And also we have measured for the surface charge. We have seen that like there is no change in the surface charge. It is once again neutral as earlier T0. Sometimes we use surface modifiers like sterile amines to enhance the encapsulation efficiency, especially for aqueous drugs, highly water soluble drugs. But in this case, there is no need for us for including any charge modifiers or charge enhancers. And with this one, like then we move to LCMS method development. And a working solution is prepared between like 10 to 1000 nanograms per ml concentration, as well as stock solutions are prepared and spiked at 550 and 100 nanogram per ml to do the partial validations. And we used famotidine as internal standard. And these dronidone hydrochloride and famotidine internal standards are injected to the rat collagen, or injected to the rat plasma, which was procured. And this is thoroughly vortex and mixed for drug solution and centrifuged at 10,000 RPM for 15 minutes. And supernatant was collected and evaporated under nitrogen flush. And we called it as protein precipitation technique. And a couple of other products, we used liquid extraction techniques, but here we used protein precipitation technique. And 100 microliters of methanol was used to reconstitute and further centrifuged, and from which 5 microliters of supernatant was transferred into inserts and then further tested for the LCMS. And coming to these LCMS conditions, we use the API 3200 model, and Zobrex C18 columns are used, particle size 5 microns, and column length is 12.5 to 2.1 millimeters. I did a couple of research on LCMS also as part of this one by taking up my lab mate Steven. And mobile phase used is given here, and mass spectrometric conditions like gas, nitrogen, and ion spray voltage. All these conditions are given here, and also it is multiple reaction monitoring mode, and also it is like a negative response mode. And given this one, we started working on the pharmacokinetic study. We have procured the Sprague Dowley rats, that is SD rats, which are pre-cannulated. You can see the pre-cannulated rats here. And these rats, 12 rats were procured, but because of the weight variation and the cannula disturbances, 2 rats were not considered. And we divided these groups into like 2 groups. Each group consists of like 5 rats for each, and fasted overnight. And we have given the pure drug suspensions, that suspension like concentration of 10 mg per ml by suspending in HPMC, hydroxypropyl methyl cellulose, also proliposomal formulation F8, and the same concentration. Time points are 0.5, 1, 2, 3, 4, 5, 6, 3, 4, 6, 8, 12, and 24 hours. And each time point we used to take 0.35 ml. Thanks to Ramana sir and Karnasri madam for letting us, getting us familiar with animal handling. Thank you very much. And also I would like to convey my thanks to Bharti madam also for becoming like more, what you say, perfect with the formulations, especially with the physical pharmacy. So all these techniques we used here, and we used to get the like blood samples, and then further centrifuge the plasma at like 3000 rpm, and insert the keeping the heparin and separate the plasma, and then add famotidine internal standard and do the protein precipitation and go for LCMS estimation. And this animal study protocol number is given here, and this is uploaded by IA Cook, WHS, Western University of Health Sciences. And finally, we came there. Yes, the pharmacokinetic study, if you see here, there is a drastic difference between the pure drug versus proliposomal formulation, which we are expecting based on all the previous studies. That means multimedia, as well as discriminatory resolution, and apparent permeability studies, that is CACO2 model, and dye studies. All these studies given us confidence and confirmation that our formulation is going to show higher concentrations definitely than the pure drug. So here both the concentrations are estimated for like plasma concentrations in nanogram per ml. And we have developed the methods, especially the analytical method as well as the LC method, in such a way that which can like quantify the nanogram levels as well. And we have done the partial validation as well, like kind of precision, intraday precisions, all those parameters we have done there. And when we see these Tmax values, plain drug is 3 hours and formulation is 2 hours. Cmax values are drastically improved, and AUC is also drastically improved from 5,000 to 7,500. So finally, we conclude that the relative bioavailability is like 148. That means almost 150%. So the study outcomes and conclusions from these studies are: dronedarone hydrochloride is an anti-arrhythmic drug having a oral bioavailability of 4%, and its physical evaluation and other parameters proven that F8 is good formulation with 84% of encapsulation efficiency and 99% of drug release at the end of 4 hours. And CACO2 has shown like 2.64-fold increase, and stability is also proven along with the DSC parameters. In conclusion, an optimized proliposomal formulation of dronedarone hydrochloride resulted in enhanced dissolution and apparent permeability compared to pure drug. The apparent permeability results were confirmed with the in vivo studies in SD rats. Also, from the pharmacokinetic data, it can be postulated that the oral absorption of dronedarone hydrochloride has been improved when administered as proliposomes. So this is the conclusions, and we have published this paper in AAPS. I'm the one of the contributing first author. I would like to thank to Priyanka Kunamanini, Stephen Eng, Nitin Swarnakar, and all my lab mates as well. So this is the research study, and I would like to quickly go through this case to study how this liposomal formulation impacts the NDA development. So in case of NDA development, we go by pre-IND, and NDA development consists of like phase 1, phase 2, phase 3 studies, which also includes of scale-up study. For this one, we have taken a model drug. This research was started in the year like 2012 and 2013. By that time, I was not part of the lab, but I joined this project in 2015. by the time we are carrying out the pre-IND studies for FDA submission for a pre-IND approval. And this is a oral drug. This is a different drug used for hypogonadism. It's a lipophilic drug with poor water solubility, and route of administration is oral route, and there are no other routes are available, especially for this drug, even for the dronedarone as well as on today. And when we see this overview, this is used for hypogonadism, and the advantages are like going by proliposons we already discussed, ease of handling, and also more stable product. And technology we use here is proliposomal formulation, that is lipophilic drug, and poorly available drug for enhancing the bioavailability. And formulation consists of phospholipids by solvent evaporation technique, cholesterol, as well as it's filled into capsule. And further capsules are given the entry coating. And technology and method of action, mechanism of action, I don't want to focus much more here. Then I would like to go to the challenges so that audience can understand like how a researcher can focus the product into commercialization and getting some patents and approvals. Especially encapsulation efficiency is very critical here. Getting encapsulation efficiency and consistency in the encapsulation efficiency is more critical when you go for scale up batches. And reproducibility, homogeneity is also more important. We have to check for the bilayer integrity, drug loading, and as well as PDA. And controls are we have to go the employ process analytical technology. We call it as PAT. And also scale up controls and also pilot studies gives us some better control over the parameters. And content uniformity also need to be taken. In case of dissolution and stability, it's a entry coated capsule. It should sustain to the acidic environment, and further it will releases after entering into the pH 4.5. Long-term stability is also proven. And in case of pre-ND, what we did is we have prepared quickly 10 to 15 formulations, and we used to check for the discriminating media, how we did for like dronedarone, and also doing the multi pH profiling with and without SLS. And we used to perform a quick animal studies. Once it passes these animal studies, that formulation was taken to the phase 1 studies. When you see these clinical outcomes, especially in clinical outcomes, we have seen a lot of values, especially in like phase 1 studies, 1A and 1B, as well as in phase 2B. As well, we have seen n number of failures. The failures are because of fluctuation of these concentrations. Other than the low solubility and bioavailability, this particular drug produces endogenously as well. So endogenous concentrations and exogenous concentrations used to change, bring variabilities, as well as because of the negative feedback mechanism, the day 1 concentrations are higher and the day 7 concentrations used to be decreased. Also, because of the harmonic variabilities, we used to see a lot of variabilities. And finally, we went to the phase 1 studies, where phase 1A, we did single ascending dose studies, and phase 1B, we did multiple ascending dose studies, and which proven the cholesterol and toxicity reduction. And oral formulations effectively achieve target serum levels at phase 1A and phase 1B. With that success, we went to phase 2, but phase 2A is required for efficacy, dose requirements, and 2B for efficacy. But the plasma levels didn't match in phase 2B studies, where we started working reformulations. I have presented this case study because to understand the audience and the researchers how to formulate how to do a research considering in the bigger picture to get kind of commercial approvals or something. And with this one, I'm concluding my topics, case studies. And thank you once again for the audience for listening for your time. And thank you once again for Ramna sir, Bharti madam, and Karnasri madam, Ravi Shankar sir, and Nagabhushnam garu, and Principal madam, Sunita madam. My special thanks to Principal madam for giving this opportunity. Thank you. Please let me know for any questions. I think we are running out of time. Yeah.
Thank you so much, sir. Thank you so much for elaborating formulation and evaluation of dronedarone formulations. Thank you so much, sir, for enlightening us with the details of all the formulation procedures and the evaluation in depth.
Thank you. Actually, there is a lot of data I thought of sharing, but I see the time is only 1 hour. Maybe next time we can take more time. Thank you.
Thank you so much, sir. Thank you so much for sparing your valuable time for us. I request Did
Any
participants pose any questions to sir? Get any clarifications?
Maybe if I think maybe I'm talking a little bit advanced. If someone is having any questions or any clarifications, also like I know that it's like actually we're running out of the time. Please feel free to reach me through my email ID. Actually, Ramna sir and other faculty is also having my email ID. If any questions are there, any discussions required, especially for planning these experiments, especially the concepts or any kind of further clarifications, confirmations, I will try to give my answers at the best. If not, I will try to check with my colleagues and even with other literature data. And definitely we can in touch and we can communicate further as
Thank you so much, sir, for the opportunity given. Can I conclude the session, sir? Can I give the closing remarks?
Definitely, but as we have 1 more minute, any questions from any faculty or something, I'm happy to I'm happy to discuss. Seems no questions, maybe. Yeah. Yes, madam, you can conclude the session. Thank you.
Thank you so much, sir. Dr. Surya Prakash Rao Kvsrscops sir has highlighted the formation and evaluation of Drondaron, an anti-arrhythmic drug formulation and evaluation procedures, both in vitro as well as in vivo procedures. Drondaron drug is an anti-arrhythmic drug working basically by blocking the multiple ion channels in heart, including sodium, potassium, and other ions. Sir has done the evaluation procedures with taking the reference product Baltac, which is developed by Sanofi Aventis, which is an NDA drug, a new drug application. Basically, the drug has a problem that it has only 4% bioavailability and is more than 95% bound to the plasma proteins. So it is a big challenge to overcome this major drawback with the drug. And Sir has developed successfully a formulation which is giving the drug release profile 99%. It's a big achievement, sir, to make a successful formulation with a drug which is having only 4% bioavailability. Sir has used materials such as DMPG, dimethyl phosphatidyl glycerol, salt derivative, which is sodium salt derivative form, then soy phosphatidylcholine and egg phosphatidylcholine, phospholipon also. And the basic method of preparation that has been used to develop proliposomes of Drondaron is full hydration method. And here the sir has suggested a keynote in the formulation development of the proliposomes of drugs such as Drondaron, that choosing the correct MWCO, MWCO, which is molecular weight cutoff, which can help actually to ensure that the drug is efficiently encapsulated within the liposomes while excess free drug is being removed effectively. So this is a keynote which has to be taken care of during the formulation of drugs such as Drondaron, proliposomal formulations. Thank you so much, sir, for elaborating with us the in detail and elaborated formulation and evaluation procedures, both in vitro as well as in vivo for Drondaron proliposomes. Thank you so much, sir. Thank you so much for sparing your valuable time for us.
Thank you. Thank you, ma'am. So, can I leave the session?
Thank you so much, sir. I have a small announcement for the participants, sir.
Yeah.
Dear participants, after today afternoon, we have one more session which is taken by which will be taken by Dr. Kvsrscops Kumar Mehra, sir, from Raipur, Hyderabad. Please join us at 3 o'clock for the 4th session of FBT program. Thank you so much. Thank you so much.
Thank you.
I'm concluding the session, sir.